From the problem presented in class, we can see that an increase in blood-free fatty acid content and a decrease in skeletal muscle GLUT4 content was observed following a 48-hour high-fat diet, but was not observed in the placebo condition.
The problem that we're looking to discuss today is how come after a 48-hour high-fat diet, there was an increase in blood-free fatty acids and a decrease in membrane GLUT4 content in skeletal muscle.
We will present and further explore this problem within the context of the conservation of mass model.
For this problem, our mass will be skeletal muscle membrane GLUT4.
Our P1 for inflow is intracellular GLUT4 content.
Our P2 for inflow is our mass, skeletal muscle membrane GLUT4.
Our conductance for inflow is RAC, which carries the GLUT4 to the skeletal muscle membrane via actin cytoskeleton.
Another factor for conductance is AS160, which when inhibited via phosphorylation allows GLUT4 to fuse to the skeletal muscle membrane.
Our P1 for outflow will be skeletal muscle membrane GLUT4, and our P2 for outflow will again be intracellular GLUT4 content. For conductance, we have not yet discussed this in class, and we do not know the mechanisms behind why skeletal muscle membrane GLUT4 is removed from the membrane.
Therefore, for the purpose of solving this problem, we will only be focusing on the factors that affect inflow.
Our conductance factors for inflow are controlled by the insulin signaling pathway. When insulin is present in the blood, it binds to insulin receptors on the skeletal muscle membrane, which ignites the insulin signaling pathway. When insulin binds to the receptor on the skeletal muscle membrane, this activates IRS in the skeletal muscle and in turn PI3K. PI3K is responsible for activation of RAC.
When RAC is activated, it transports GLUT4 to the membrane along the actin cytoskeleton. PI3K further activates the insulin signaling cascade through activation of AKT and PKB, which in turn inhibits AS160. This inhibition of AS160 allows GLUT4 to be translocated to the skeletal muscle membrane.
What mechanisms might exist that link the decreased skeletal muscle membrane GLUT4 content and the increased blood-free fatty acid content that we see in our problem?
We know that with a high-fat diet comes an increase in blood-free fatty acids. This increase in free fatty acids is our P1 and creates a greater concentration gradient for inflow of blood-free fatty acids into our skeletal muscle through membrane transporters.
Once inside the muscle, free fatty acids are converted into fatty acyl-CoA. From here, fatty acyl-CoA can be converted either into ceramides, which is one type of reactive lipid species, or can undergo reesterification.
The process of reesterification takes fatty acyl-CoA to LPA, then to DAGs, which is another reactive lipid species, and eventually into TAG.
Due to an increase in inflow of blood-free fatty acids into the muscle, we would expect to see an increase in all intermediates, including our reactive lipid species, DAGs, and ceramides.
Both DAGs and ceramides inhibit the insulin signaling cascade. Ceramides inhibit AKT and PKB, while DAGs inhibit IRS. Inhibition of IRS results in an inhibition of PI3K, which leads to an inhibition of RAC. Of AKT and PKB results in a decrease in the phosphorylation of AS160, also known as activation of AS160. With the decrease in phosphorylation of our conductance factors, RAC and AS160, we would expect to see a decreased translocation of GLUT4 from the cell to the skeletal muscle membrane.
Therefore, we hypothesize that skeletal muscle membrane GLUT4 content is reduced following an increase in blood-free fatty acid due to increases in reactive lipid species, for example, DAGs and ceramides. To determine if our hypothesis is plausible, we looked at a study by you and colleagues. The purpose of the study was to explore the possible roles of different intracellular fatty acid metabolites, such as fatty acyl-CoA, DAGs, ceramides, and triglycerides, in mediating fatty acid-induced insulin resistance in skeletal muscle. In order to test the effect of these fatty acid metabolites, male Wistar rats were matched in weight and then randomly allocated into 5 different lipid infusion groups after an overnight fast. Blood free fatty acid concentration increased rapidly following the lipid infusion and remained constant until the saline washout period. At the end of the infusions, rat muscle samples were taken and frozen. DAGs and ceramides were extracted from frozen tissue and then analyzed. The results show that this increase in blood-free fatty acid concentration resulted in increased intramuscular DAG concentrations in the soleus muscle compared with the controlled group.
These results are illustrated in Figure 1C of the paper by Yu and colleagues. Yu and colleagues also found that there was no change in intramyocellular ceramide content following lipid infusion.
This can be shown by Figure 1D in the study. Importantly, the study also observed that following an increase in intramyocellular DAG mass resulting from elevated blood-free fatty acid, there was also a 30% reduction in insulin activation of IRS-1 tyrosine phosphorylation and approximately a 50% reduction in IRS-1-associated PI3K activity.
So the literature that we found supports that DAGs do have an influence on the insulin signaling pathway, but it found that ceramides do not. This effect on IRS that DAGs has causes a decrease in the conductance for our mass, which is skeletal muscle membrane GLUT4 content. Therefore, we can generally accept our hypothesis that skeletal muscle membrane GLUT4 content is reduced following increases in blood-free fatty acids due to an increase in reactive lipid species. However, the literature does not show an increase in ceramides, so we can assume that DAGs play a more important role.
This explains why we see a decrease in skeletal muscle membrane GLUT4 content in response to increased blood-free fatty acid levels, which is why eating a high-fat diet can rapidly alter insulin sensitivity and glucose uptake in skeletal muscle.